Hey students, just wanted to create this quick video of the lesson that we did this morning in case you need to go back and rewatch any portion of it. So in this lesson, number 3, we did two main things. I spent some time just describing what's known as simple Mendelian genetics, and then I spent some time thinking and talking about examples of advanced or non-Mendelian inheritance genetics. So first, let's talk about simple Mendelian inheritance. And what this is referring to is essentially back to Mendel's primary experiments with pea plants, right, where there were multiple traits he was considering, such as seed shape or flower color, and he determined through various crosses the dominant form and the recessive form of the trait, and was able to, through establishing things like the law of segregation, the law of independent assortment, was able to understand these crosses and also make very precise predictions about the ratios of the offspring. And so when we talk about simple Mendelian inheritance, we're talking about traits that are controlled by one gene, and for that gene there is a dominant and a recessive allele. An allele is just referring to the different versions of that same gene and ultimately the traits that that gene controls. All right, and this goes along with exactly what he was observing. Many of these traits, such as flower color, are controlled by one gene, and there's a dominant form, which was the purple, and a white form, which was the recessive allele. Simple Mendelian inheritance is also referred to as meaning we get, or the offspring receive, one allele from each parent, and the specific combo of alleles, which we call a genotype, leads to either the dominant or the recessive phenotypes. Right? So again, this is exactly what Mendel observed and explained, and we're referring to traits that follow the law of segregation, which is what we just wrote here, is that for the trait, we receive one allele from each parent, and it's random which allele is passed down for each parent. Follows the law of segregation and the law of independent assortment, which means it does not, the inheritance of a particular allele for a trait does not impact the inheritance of an allele for a different trait, right? They're independent events. Okay, so an example of a simple Mendelian genetics problem would be something like this. So we looked at this in class that two cats are mated. One of the parent cats is long-haired, which we learned here is the recessive allele. The litter, which results, contains 2 short-haired and 3 long-haired kittens. What does the second parent look like, and what is its genotype? So let's start with just writing down the long-haired or short-hair phenotypes correspond to, for the long hair, this is the recessive form of the trait, and short hair is the dominant version of this trait. So I'm just going to use the letter A. It doesn't matter what letter you use, it just matters if it's uppercase or lowercase. So the recessive allele is always represented by a lowercase letter, and a dominant allele is represented by an uppercase letter. And so what we learn is that we've got parent number 1, who is long-haired, and we're trying to determine parent number 2. We don't know the phenotype and we don't know the genotype yet, but we learned that the offspring, there are 2 short-haired kittens and 3 long-haired kittens. Okay, so then let's start thinking about genotypes. So first, since the genotype, or since the recessive version of this trait is long hair, that's only going to show up as an individual's genotype if they have a homozygous recessive genotype. So parent number 1, who has long hair, must have the genotype lowercase a, homozygous recessive. And all of the offspring that have long hair also should have homozygous recessive genotypes. Now, the short-haired offspring, since short hair is the dominant, that just means that they have at least one dominant allele. So they can either have what we call homozygous dominant or a heterozygous genotype. But as we look at this problem, we can actually determine that the kittens that are short-haired are likely to be heterozygous. In fact, they have to be heterozygous. And here's why. So we learned that parent number 1 is homozygous recessive. And so for every offspring, each parent's going to contribute an allele, which means that every offspring is going to receive a recessive allele from parent number 1. And so it's impossible to have a homozygous dominant genotype since the offspring will always receive at least one recessive allele. So then we can think about parent number 2. So parent number 2 must have at least one dominant allele, because the dominant allele that's leading to these short-haired kittens can't be coming from parent number 1. It has to be coming from parent number 2. And because this set of parents also had some long-haired offspring who are homozygous recessive, this parent number 2 must also have a recessive allele, right, in order to account for the fact that these offspring down here had to have received one recessive allele from parent number 1 and one recessive allele from parent number 2. So we know that parent number 2 now is heterozygous as the genotype. And what is the phenotype? Well, since they have one dominant allele, this must mean parent number 2 is short-haired. Okay, so that's just an example of a simple Mendelian genetics practice problem involving one trait controlled by one gene with a dominant and a recessive allele. Okay, so let's think about examples of traits that follow what we call advanced or non-Mendelian inheritance patterns. And so in class I mentioned that there are lots of different examples, but really what you need to know for this class and for the AP exam is just a little bit about each one of these and an example of each one of these. So let's go ahead and think about a few of these on this slide. So first, an example of a trait that maybe doesn't follow simple Mendelian inheritance would be a trait with incomplete dominance. And the major example here is actually flower color in snapdragon flowers. This is where we primarily think about this idea of incomplete dominance. And what this means is that in the case of a heterozygous genotype, right, in normal or in simple Mendelian inheritance, the phenotype of the heterozygous and the homozygous dominant offspring will all be the same, since they each have at least one dominant allele and therefore they'll show the dominant phenotype. But in incomplete dominance, this is characterized by the heterozygous individuals having what we call an intermediate phenotype. And so because the allele for red flower color in snapdragons isn't fully dominant, and the allele with regards to white flower color isn't fully recessive, they actually use different letters. They use R and W. They don't follow the kind of convention of uppercase and lowercase. And so you will only see that full red color in the case of a homozygous genotype with 2 copies of that red allele. And similarly, you will only get white flower snapdragons if you have homozygous for the white allele. But here we see the intermediate phenotype, that's pink, right, illustrating this idea of incomplete dominance. Okay, the second example of advanced or non-Mendelian inheritance is this idea of codominance. And the idea here is that there are some traits where there are multiple dominant alleles. And probably the main example that we need to be familiar with is blood type, right? And you've likely heard that there are blood types such as type A, B, AB, or O. And then you've also maybe heard about positive and negative, like A positive or A negative. The gene that actually controls whether or not we have a positive or a negative blood type actually does follow simple Mendelian inheritance patterns. It's controlled by one gene. There's a dominant and a recessive allele. But for the other part of blood type, A, B, and O, there are actually multiple dominant alleles that someone can have that will be expressed as their phenotype. And those alleles are represented as the superscripts on the letter I. So with I superscript A representing the allele for A blood type, I superscript B corresponding to B blood type, and lowercase is the actually the recessive allele that does not correspond to either A or B. So we say that the alleles for A and B are both dominant. They're codominant alleles, meaning that if a person has A and B, they'll both be expressed. Well, this is our recessive. Now, this gets down to really how this blood type is established. Essentially, these genes with the alleles IA or IB, they determine whether or not a particular enzyme is produced in the body that adds this particular carbohydrate marker on the surface of our red blood cells. So someone that has the allele A or IA, in either of these two forms, the red blood cells will have this A carbohydrate indicated by this yellow triangle on the surface, and similar for type B. However, in someone with type AB blood type, that person is producing both enzymes that add both carbohydrate A and carbohydrate B to the surface of those red blood cells, and so therefore you get both phenotypes expressed on the surface of the red blood cell. And type O is simply where both of those enzymes are not present, and so therefore you get neither of those carbohydrates on the surface. Okay, so that is codominance. And our major example, again, is blood type. So let's talk about this third example here of advanced or non-Mendelian inheritance. This gets down to polygenic inheritance. And when we think about this, some major examples of polygenic inheritance are things like skin color, hair color, or height. And the idea is that there are multiple genes controlling the trait. And the reason this is important is because this allows for what we call quantitative traits. And what that means is that, for example, skin color or hair color or height we know that there's not just one or two types of skin color, hair color, and height. There's actually a continuum, right, for each of those types of traits. And so the way that's accomplished is actually, for example, in this figure, this is for skin color, there are multiple genes, in this case 3, with 2 alleles each, that correspond to a person's skin color. And the specific combination of dominant and recessive alleles will actually result in lighter or darker skin pigmentation. So this allows for more of a gradient when you have multiple genes contributing to a particular trait. Okay, so that's incomplete dominance, codominance, polygenic inheritance. Let's talk about a few more here. So another example is something called phenotypic plasticity. Phenotypic plasticity. And the idea here is that there are traits that are controlled by both genetic and environmental factors. And by trait here, I'm referring to kind of the phenotype for a particular trait. Right, and what this means is that take, for example, skin color. We know that skin color is impacted by genetic elements, but also environmental elements as well, such as UV exposure can actually increase the skin pigmentation for a particular individual because your body actually ramps up a type of melanin production in the presence of more UV. What we also see this is in a particular type of flower called hydrangeas, and hydrangeas have this trait of flower color that not only is dictated by certain genes, but can also be affected by the chemistry of the soil, with more acidic soil compositions leading to more deep blue colored hydrangea flowers, and soils that have a pH closer to neutral or 7 having closer to a pink or deeper pink color. And then lastly, we see here there are on the bottom of a leaf of a plant, they have these valves of sorts called stomata that actually allow for gas exchange, for example, in the process of photosynthesis. And not only is the amount of stomata present on a leaf dictated by genetic factors, but also the amount of carbon dioxide present in the surrounding environment can influence how many stomata are on that leaf. This is also related to what we see research surrounding mental health disorders is looking into, is something like depression that can have both genetic and environmental factors that can play into the presence of depression. Okay, so that's phenotypic plasticity. The next example here is something called non-nuclear inheritance, or extranuclear inheritance is another name. And the main idea here is that we learned back in the beginning of the school year, Unit 2, about the theory of endosymbiosis, and in that theory we learned that these two organelles, mitochondria and chloroplasts, which both are present in plant cells, but only mitochondria are present in animal cells, they actually contain their own DNA. And it's found that the genes that are contained within that DNA are inherited in a non-Mendelian fashion. In fact, they're primarily inherited maternally, because actually the egg for animal reproduction provides the entire amount of mitochondria that will then divide and be spread amongst the daughter cells as that zygote divides after fertilization. So actually all of the DNA from the mother's mitochondria will be passed along and none from the father. And similarly, we see that in plants with the ovum as well. Okay. The last thing that we're going to talk about in this lesson of an example of non-Mendelian inheritance is something called sex linkage or sex-linked traits. And the idea here is that, remember when we introduced chromosomes, right? In humans, we say that the first 22 chromosome pairs are autosomal. They're autosomes. And the last pair are our sex chromosomes. And on each of these chromosomes, there are mini genes, right, that control traits. And typically, when we refer to simple Mendelian inheritance patterns, we're usually referring to traits that are controlled by a gene located on autosomes. Because you see, with autosomes, you're always guaranteed to receive 2 alleles for any particular gene because you've got 2 copies of each chromosome, right? But for sex chromosomes, because biological female and biological males inherit different combinations of sex chromosomes, this can result in sometimes only receiving 1 allele for a particular gene or 2. Most of the time when we refer to sex-linked traits, we're talking about traits controlled by genes found on the X chromosome, simply because it's a larger chromosome. Many more genes are found there. Some examples that we're going to see are things like hemophilia, a genetic disease that leads to the phenotype of excessive bleeding or the inability to form blood clots, or color blindness as well. Both are caused by mutations found in genes on the X chromosome. And because a biological female will receive 2 X chromosomes and a male will receive 1 X chromosome and 1 Y chromosome, if the gene is found on the X chromosome, that means a male will only receive 1 allele for that gene, whereas a female would receive 2. And that's going to introduce different inheritance patterns in comparison to what we would see with simple Mendelian inheritance. Okay, so that was what we went over in class. I hope that if you missed anything, you were able to go back and rewatch this. But I hope you have a good rest of your afternoon.